Hello, I'm Danny Cucciara. My presentation is about anthrax caused by the pathogen Bacillus anthracis. I noticed going through ProMed Mail that many of the anthrax outbreaks that included humans were related to consumption of dead infected animals, even when this was against public policy or advice. I will give you several examples, but first I think it's important to understand a little bit about the disease and the pathogen that causes it. Anthrax is a bacterial zoonotic disease, meaning that animals can get it and can pass it to humans, but humans can't pass it between each other. It is typically spread to humans after they have handled diseased animals or animal products. There are around 2,000 cases per year worldwide of anthrax. Those cases are rare in developed nations because farms in developed nations can afford to vaccinate their livestock against diseases like anthrax. So most outbreaks are in agrarian regions and in developing nations, places where livestock may be the lifeblood of the people who live there. People whose lives and livelihood are closely dependent on their animals can be reluctant to destroy a sick or dead animal when that animal can be used to feed their family or their community. This situation, as I will show in this presentation, happens commonly even in places where public health officials try to educate the population about anthrax infection. Anthrax is emerging in the US and Europe over the last 20 years. In Europe, there have been outbreaks due to tainted heroin use. It is suspected that the heroin came from areas where anthrax is endemic. So injectable anthrax is an emerging concern in those affected countries. In the US, since the 1990s anthrax attack, there is a concern that anthrax may be used as a bioweapon. Mortality rates vary with infection rate and the amount of time it takes for an infected person to seek treatment, among other things. Those who are treated in a timely manner have a high likelihood of survival. Untreated infections generally have about a 20% mortality rate, while treated infections have a much better prognosis. 95% of all infections are cutaneous, meaning the root of infection is via the skin, normally through an opening such as a wound. This is the bacteria responsible for the infection. Bacillus anthracis is a large, rod-shaped, gram-positive bacterium with a single polar flagellum. It lives as an endospore in soil outside of a host, though there is some evidence that B. anthracis can promote plant growth and may be active in nutrient-rich, bloody soil. This is interesting because historically it was believed that anthrax was dormant in soil until encountered by a host. Once ingested by a host, the bacterium moves through the blood or lymph to a lymph node. The bacteria attacks extracellularly, but the toxins work in the cytoplasm of the cell. Its primary virulence factors are its peptidoglycan cell wall and anthrax toxin, which I will discuss in detail in a moment. Anthrax toxin is an important aspect of treatment because the buildup of toxin could be fatal even after the bacteria have been treated by antibiotics. B. anthracis life cycle takes place in two forms: the endospore and a vegetative cell. The bacteria will sporulate or germinate depending upon available nutrients. Typically, it sporulates as a deceased animal decomposes on the soil. As the nutrients are used up, the bacterium becomes a spore to await its next host. The spore is made up of genetic material and a super thick multi-layer coat, as you can see here. It's resilient and can withstand very inhospitable conditions, such as high heat or UV radiation. Once ingested by a host, or when nutrients become available, the spore will germinate into a vegetative cell. The bacteria enter the body as a spore via the infection route. Once in the body, macrophages are alerted to the invader's presence. The macrophages exit the bloodstream and engulf the bacteria via phagocytosis. The bacteria survive this because of all of the protective cell wall encasings. Indeed, the bacteria germinate within the macrophage. The macrophage enters the bloodstream and travels to a nearby lymph node. Once in the lymph node, the bacteria reproduce and escape the macrophage. They attack lymph cells and travel along the lymphatic system to access other parts of the body. As the bacteria attacks cells, they produce virulence factors, such as anthrax toxin. This damages the cell's ability to regulate itself, and the cells produce water. This is known as edema. In respiratory anthrax in particular, it is known as pulmonary edema, when the lungs fill up with water. Edema can cause significant tissue and organ damage. Anthrax toxin and the bacteria's capsule are encoded by PXO1 and PXO2, which is regulated by the ATXA transcription factor gene product. Anthrax toxin is made up of 3 proteins, a protective antigen, and 2 toxin factors, lethal factor, which is coded by PXO1, as is edema factor. PXO2 encodes for the capsule. Protective antigen binds with ATR, which is anthrax tumor receptor, and which is encoded by tumor endothelial marker 8 gene. Or it can also bind with another receptor encoded by capillary morphogenesis gene 2, CMG2, on the host cell wall. Both of these receptors are common in many kinds of tissue. The protective antigen is made up of 2 subunits. Furin cleavage releases the smaller subunit PA20, then subunit PA63 becomes a PA monomer. The monomer becomes a heptamer and forms a prepore on the host cell wall. As many as 3 lethal factors or edema factors can compete to bind with the protective antigen, heptamer. Once bound, the complex undergoes endocytosis using a pH gradient to enter the cytoplasm. Here, the complex releases the lethal factor or edema factor. Edema factor increases intracellular cAMP levels via, I'm going to try and pronounce this, so be patient with me, calmodulin-dependent adenyl cyclase activity. Lethal factor inhibits mitogen activated protein kinase 1 to 4, 6, and 7. Both lethal factor and edema factor are known to influence the function of several different types of host cells. Edema factor becomes an adenylate cyclase, which helps to catalyze abnormal production of cyclic adenosine monophosphate, cAMP. cAMP can alter ion and water movement, resulting in edema. Edema toxin is thought to disrupt signaling pathways, damage the ability of neutrophils to function, and it can disable phagocytes and cytokine pathways. Edema toxin may play a role in inflammatory processes and shock. Lethal factor becomes a zinc-dependent protease that cleaves the amino termini of 6 different mitogen-activated protein kinases, or MAPKK. This causes disruption of the regulation of molecules through phosphorylation cascades. It affects pathways involved in cell growth, maturation, and cellular stress response. Lethal toxin can impair the ability to initiate innate and adaptive immune responses. It does this by interfering with cytokine IL-8 mRNA and other kinds of cytokine gene expression. This disruption of the MAPKK pathway can lead to blocked stress response and apoptosis in vascular endothelial cells. It is thought that lethal toxin plays a role in shock. How is it spread to people? The soil is the primary reservoir for B. anthracis. It can remain in the soil as a spore for decades, maybe longer, waiting for a host to come along. Animals, or potentially insects, inhale or ingest spores while grazing. Interestingly, research has found B. anthracis in the guts of flies and mosquitoes, so the potential exists that they may be a vector or a reservoir. Infected animals die very quickly if they aren't vaccinated against anthrax. As the corpse decomposes and the bacteria move back into the soil, sporulate as nutrients are used up and wait for another host to come along. The infection can be passed to humans who work with infected animals or animal products. Also, it can be passed to humans who consume undercooked meats from infected animals. People who are at risk for anthrax infection are those who could potentially come into contact with unvaccinated animals that are infected, like farmers or herders or veterinarians. Anyone who works with animal products. An example of this would be sheep's wool or people who tan hides to make leather. Those who travel to countries where anthrax infections are known to occur are at risk. Scientists who study the bacteria are at risk. Military personnel. As a soldier, I was vaccinated against anthrax before I deployed to the Middle East in the early 2000s. Mail carriers, as with the bioterror attack in the US during the early 90s, and those who work to fight bioterrorism are also at risk. There are 3 typical anthrax transmission routes. The most common is cutaneous infection. Most often this occurs on an exposed area of skin like the face, arms, hands, or neck. The infection accesses the blood or lymph stream via abraded skin or an open wound. 95% of anthrax cases are cutaneous. With treatment, the mortality rate is about 1%, but without treatment, it is anywhere between 5 and 20%. The most dangerous type of infection is respiratory or pulmonary anthrax. This type of infection occurs when spores are inhaled. Spores travel through the respiratory system into alveolar sacs where they attack. Inhaled anthrax has a 46% mortality rate untreated. The rarest infection route is gastrointestinal. This is when infection occurs because of spores or bacteria ingested in water or undercooked meat. Gastrointestinal anthrax can be oropharyngeal or intestinal, depending on where the bacteria settle. Untreated gastrointestinal anthrax has a mortality rate of 25 to 60%. Injectional anthrax is similar to cutaneous. Though rare, it has a mortality rate of 33%. Though this infection type can be dangerous because the bacteria are injected directly into the bloodstream, and the possibility that people who are injecting illegal drugs are not necessarily as healthy as non-drug users to begin with. Because infection routes, bacteria loads, and the health of affected individuals vary, incubation time is also variable. It can be a day to several weeks until spores germinate and symptoms appear. Symptoms are similar for all 4 types of infection. All forms may cause flu-like symptoms such as nausea, muscle aches and pain, swelling lymph nodes, fever, vomiting, possibly shock and septicemia in the worst cases. In cutaneous and injectable, there will be swelling and bumps or blisters at the infection location. These bumps may be itchy. Eventually, as the blisters or bumps go away, an eschar will appear. This is a sore with a black scab topping. Nearby lymph nodes may swell, and there can be edema or necrosis. Shock and death are rare in cutaneous infections. In addition to the common symptoms, respiratory anthrax can cause cough, shortness of breath, dizziness, sweating, and exhaustion. Meningitis is more common in respiratory anthrax as well. Gastrointestinal anthrax, besides the common symptoms, there might also be flushing of the face or eyes, swelling of the stomach, bloody vomit, diarrhea that can also be bloody. If the infection is oropharyngeal, there can be glandular swelling in the neck. There may also be sore throat and pain when swallowing and hoarseness. This type of anthrax can also result in shock and meningitis. There are many ways to diagnose anthrax infection. I noticed in my research that it is often done using PCR to identify B. anthracis genes. When PCR is not possible, they can test fluid from lesions, blood, throat swabs, stool, and spinal fluid for the bacteria. They test the spinal fluid when they suspect meningitis due to anthrax infection. Chest X-ray can be done to detect respiratory anthrax. Anthrax is normally treated with antibiotics such as Cipro or doxycycline, though, as I have mentioned, eliminating the bacteria may not be enough to save the life of the infected. Antitoxins may be used if the toxin levels are high. In many cases, once an outbreak has been discovered, those at risk will receive vaccination. The vaccine is normally intended for those who are at an elevated risk for the disease, such as people who live in an outbreak area or work with unvaccinated animals that are at risk. Here is a list of food safety-related anthrax outbreaks that occurred within the last 12 months since April 2018. This is by no means an exhaustive list of anthrax outbreaks. It is only a few cases that were confirmed linked to food safety breaches. In India, there have been 4 people infected with anthrax. The previous year in September, there was a similar outbreak in the area that infected 5 people. This month in Zhonghui City in China, 3 people were infected and 1 of those people died from anthrax infection. In Turkey, 73 people were hospitalized with a suspected anthrax infection. 1 of those people died in that outbreak as well. In Uganda, there is a refugee situation that has exacerbated the anthrax outbreak occurring there this month. 2 people were infected and 1 person died of the infection. In Thailand, 4 people contracted anthrax after slaughtering infected goats that were smuggled across the border from Myanmar. Over 30 people ate the meat from these goats but did not appear to be infected. In Kyrgyzstan, an infected cow was slaughtered and eaten, which resulted in the hospitalization of 3 people for anthrax infection, and 15 other people were listed as under medical supervision. In Guinea, 5 people were confirmed to have contracted anthrax and 1 died of it. Guinea. These cases are linked to an infected cow. In all of these situations, infected livestock died, were prepared and eaten. Many areas have a public health policy that covers how infected animals are handled. Typically, infected animals should be euthanized. Corpses should be burned or buried significantly deep to prevent infection from spreading. This is very hard to enforce, especially in areas of low socioeconomic status. Often, survival in these areas is closely linked to their livestock. An outbreak has many repercussions besides illness when a family or community depends on a herd of animals for their livelihood. It is also difficult to justify expensive vaccination if an outbreak hasn't happened in that area for a while. Often, when an animal dies of anthrax or for any reason, it is butchered and eaten. This is justifiable because it means that a valuable animal won't go to waste. This failure to properly handle the carcass often results in cutaneous anthrax infection in people who prepared the meat. It can also result in bacterial infection in the soil where the carcass was prepared. If the meat isn't fully cooked, it can result in gastrointestinal anthrax. This is why it's important to test people even when they have not shown signs or symptoms of the disease but have eaten the meat. These behaviors are culturally bound and very difficult to change. There is a great need for repeated intervention and education about the need to vaccinate livestock and destroy the corpses of infected animals. Thank you for listening to my presentation.